What if your muscle could tell you when your training intensity is about to change? Not your heart rate, not a blood lactate sample, not only your breathing, but the oxygen signal inside the working muscle. In this video, we are looking at the article, Assessing the Feasibility of Near-Infrared Spectroscopy for Evaluating Physiological Exercise Thresholds. It was published in Scientific Reports by Iskra and Paravlić. The study tested whether NIRS, or near-infrared spectroscopy, can help detect training thresholds during an incremental cycling test. These are the thresholds coaches use to define endurance zones, pacing, and training intensity. And the key question is practical. Can a muscle oxygen sensor help identify VT1 and VT2 without relying only on lab gas analysis or blood lactate? Before we continue, make sure to subscribe to Sports Science Insight. Here we break down sports science research into simple, practical, and evidence-based insights for athletes, coaches, students, and practitioners. Let's break it down. To understand why this study matters, we need to start with thresholds. In endurance training, thresholds help define intensity zones. Below the first threshold, the effort is usually more sustainable. Around the first threshold, the body begins shifting toward higher metabolic stress. Near the second threshold, the athlete approaches a much harder intensity domain. Above that point, fatigue rises quickly. That is why VT1 and VT2 are so important. They are not just numbers from a lab report. They help answer practical training questions. How easy is easy? Where does tempo work begin? Where does threshold training become too hard? And when does the athlete enter a severe intensity zone? Good endurance training starts with knowing where intensity changes. And this is exactly where NIRS becomes interesting. Traditional threshold testing is powerful, but it is not always practical. Gas exchange analysis can detect ventilatory thresholds with strong physiological meaning, but it requires a metabolic cart, a mask, laboratory conditions, calibration, trained staff, and time. Blood lactate testing is also useful, but it requires repeated blood samples, consumables, and it cannot measure continuously. So the problem is not that these methods are bad. The problem is access. Many coaches, athletes, and teams want more practical ways to monitor intensity. That is why wearable technologies are attractive. A NIRS sensor can be placed over a working muscle, such as the vastus lateralis, and track how oxygenation changes as exercise intensity rises. The challenge is clear. Can we make threshold testing more accessible without losing accuracy? That is the promise this study tested. So, what does NIRS actually measure? NIRS uses near-infrared light to estimate oxygenation changes in the muscle. In this study, the sensor was placed on the vastus lateralis, a key muscle during cycling. The researchers analyzed several signals. O2-HB, or oxygenated hemoglobin, reflects oxygen carried in the local muscle region. HHB, or deoxygenated hemoglobin, increases as the muscle extracts more oxygen. THB, or total hemoglobin, gives information about local blood volume. And TSI, the tissue saturation index, reflects the balance between oxygen delivery and oxygen utilization. As cycling intensity increases, the muscle needs more oxygen, so the oxygenation pattern changes. This is the key idea. NIRS shows how the working muscle handles oxygen as intensity rises. It gives a local muscle view of effort, not just a whole body view from the lungs. The study included 27 male cyclists. They were divided into two groups. One group included highly trained endurance cyclists. The other included recreational cyclists. Everyone completed a step-incremental cycling test to exhaustion. The test started with a warm-up at 100 watts. Then, intensity increased by 20 watts per minute until the athlete could no longer continue. During the test, the researchers measured gas exchange to determine VT1 and VT2. At the same time, the NIRS sensor measured muscle oxygenation on the vastus lateralis. Then, the researchers looked for breakpoints in the NIRS signals. The key comparison was simple. Do muscle oxygenation breakpoints happen at the same intensity as ventilatory thresholds? This matters because if they do, NIRS may become a practical complement for endurance testing. Now comes the main result. The NIRS-derived thresholds did not differ significantly from the ventilatory thresholds. That means the breakpoints detected from muscle oxygenation aligned with VT1 and VT2. The correlations were also meaningful. For thresholds around VT1, correlations were moderate to strong. For thresholds around VT2, correlations were even stronger. This is important because VT2 is often one of the key markers for high-intensity endurance prescription. In simple terms, when breathing patterns showed a threshold, the muscle oxygen signal showed a related breakpoint too. That does not mean NIRS perfectly replaces gas exchange testing, but it does mean NIRS can capture useful physiological transition points. The key message is this: muscle oxygenation breakpoints aligned with ventilatory thresholds. That is a strong signal for practical use. The study looked at both HHB and TSI, and here, TSI looked especially promising. TSI showed lower variability than HHB. That matters because coaches need signals they can interpret consistently. A sensor is not useful if the data are too noisy or too hard to explain. TSI followed a clearer response pattern during the incremental test. At lower intensities, the signal was relatively stable. As intensity increased, TSI declined. Then the curve showed breakpoints that could be identified using segmented regression. This matters because those breakpoints may correspond to training thresholds. HHB can also be useful, but in this study, its response was more variable. Some participants showed 2 breakpoints, some showed 1, and a few showed none. So for real-world use, TSI may be easier to interpret than HHB. That makes it a practical candidate for coaches and sport scientists. The study also compared highly trained cyclists with recreational cyclists. And this is where the physiology becomes really interesting. The highly trained cyclists had a higher VO2 peak. They reached thresholds at higher oxygen uptake. They also achieved VT1 and VT2 at higher relative intensity. That is expected, but the NIRS data added another layer. The highly trained cyclists showed a tendency toward lower minimal TSI values. In simple terms, their working muscles appeared to reach deeper deoxygenation during the test. That suggests better peripheral oxygen extraction. Their muscles were able to use more oxygen during hard exercise. The study also found a moderate negative correlation between VO2 peak and minimal TSI. So athletes with higher VO2 peak tended to show lower muscle oxygen saturation at maximal effort. This suggests that elite endurance training may improve how muscles extract oxygen. Not just how much oxygen the lungs and heart can deliver. But we need caution. NIRS is promising, but it is not magic. Sensor data only becomes useful when the method behind it is reliable. First, sensor placement matters. The device must be placed over the correct muscle belly and secured properly. Second, signal processing matters. Motion artifacts, noise, and outliers can distort the curve. Third, the breakpoint detection algorithm matters. Different methods can produce different thresholds. Fourth, the protocol matters. Step duration, intensity increases, warm-up, cadence, and test design can all influence the response. So we should not assume that every wearable sensor automatically gives valid training zones. A device alone is not the method. The key idea is this: a sensor is only useful when the protocol, placement, and analysis are reliable. That is why standardization matters. So what should coaches and athletes take from this study? First, NIRS-derived parameters can help assess exercise thresholds. Second, TSI may be more practical than HHB because it showed a cleaner and less variable response. Third, NIRS can provide muscle-specific information that traditional gas exchange does not show directly. Fourth, highly trained cyclists may show different muscle oxygenation responses than recreational cyclists. Fifth, NIRS should be used as a complement, not as a magical replacement for all testing. The real value is context. Gas exchange tells us what the lungs and whole body system are doing. NIRS tells us what the working muscle is doing. Together, they can give a stronger picture of endurance physiology. The bottom line is this: NIRS may help coaches see what the muscle is doing, not just what the lungs report. If this video helped you, leave a like, subscribe to Sports Science Insight, and stay tuned for more evidence-based content on endurance training, physiology, recovery, and performance technology. And tell me in the comments, would you trust a muscle oxygen sensor to guide your training zones? Or would you still prefer lab testing with gas exchange or lactate? See you in the next breakdown.